Okay, so I'm going to, which one? Okay, good. This just is disclaimer, which has to be shown here. There's no time to read these things, but it has to be shown when you're coming from a company. So I'm going to walk you through the Lyme borreliosis vaccine that we have developed in our company. So Lyme borreliosis is then caused by Borrelia, and the family of Borrelia the Lyme borreliosis family is composed of 14 different species, at least, might be more now, was always increasing, but there are only 4 species causing the majority of the clinical cases. And this is an odd bacteria, has no LPS, instead it has a lot of lipoproteins on the surface, growing very slowly and spiral shaped. You can see it very long, elongated, has a segmented genome, 1 linear chromosome, and a lot of different plasmids, both linear and circular. And these are then essential for the virulence and the in the vertebrate and ticks. And there are a lot of uncharacterized genes. The different Borrelia species are serotyped based on the OspA protein on the surface. And there's a low homology between the species, and within one of the species, Garinii, we have different serotypes. So very homogeneous protein. And this is the protein that we are basing our vaccine on. And it's only transmitted by exodes ticks. And here you can see the different stages of ticks. Up here is a small dot, that's a larva, the first stage. And down here we have a fully fed adult. And here we can see then the adult, the pregnant woman is then, female, is then laying more than 1,000 eggs. And these are then the start of the whole cycle that's going through. It's not that good. The battery's out, or yeah, okay. It starts with eggs that's molting into larvae. The larvae is feeding. The larvae are not infected with Borrelia because there's no transovarial transmission of Borrelia. So they're picking up the Borrelia in the first feed by small mammals, rodents, or birds. They are then falling off after feeding 3 to 5 days. They're molting into the nymphs. Nymphs are normally the tick stage that are infecting the humans or attacking humans. And they are going on all kinds of animals. They are then falling off after a week, molting into adults. The adults are then feeding on larger animals because they can't feed on small rodents, because they would be anemic, because they are sucking up to 2 milliliter blood, and that would be too much for a mouse. And then they are mating on the mammal that they are feeding on, and then falling off, and the female is then laying the eggs, and then the cycle starts again. And the cycle usually takes 2 to 3 years to go through, depending on the climate and where it is. The Borrelia and Lyme borreliosis only exist in the northern hemisphere, and you can see it in US, both the west coast and east coast, and nothing in between. And there in US, there's only one serotype or one species, Borrelia burgdorferi, with a serotype 1. In Europe and Asia, all the way to Japan, we have all 4 species, and there can also be then up to 7 serotypes. And if you're looking, we have here representing 2 different strain collections, one from a German collaborator, Volker Fingeler, and one from our competitor Baxter. And the left one, there's only strains human isolate from Germany. And the most common strain is the Borrelia afzelii, OspA serotype 2. And from the Baxter collection is from 16 different countries, mainly Hungary, but all other, so Scandinavia also. And we see in both cases the serotype 2 is the most common. And if we take the 6 most common serotypes, that should cover more than 95% of all human infections. And just to show you the underrepresentation and the progression of the Lyme borreliosis. And here we have a map from US from 2001 on the left side and 2011 on the right side. And you can see the blue dots represent cases of Lyme borreliosis. And you see how it's increasing. And earlier this year, CDC was publishing a new estimate about the number of cases. Before it's been between 20 and 30,000 per year. Now they estimate it to be almost 300,000 cases per year. So even in US, a reportable disease is underreported. And now with the new estimate is the third most common reportable disease in US after chlamydia and gonorrhea. And even if you go for the northwest, New England and that's the second most common. And just show you the disease is caused by the Lyme borreliosis. What most people know is erythema migrans, the red spot you get around the tick bite. And this is not actually what we want to prevent with the vaccine, because when you spot this, you can easily treat it with antibiotic. The problem is that not everyone gets the red spot and not everyone notices, especially with kids. If you get a tick bite on the head, because of all the hair, you don't really see it. And then it can go over to a disseminated infection where the borrelia moves to joints, nervous tissue, or heart. And there you have the problem then to treat it, because you need longer courses of antibiotic or higher doses. And also in US, there are some cases from death reported from heart failure caused by Lyme borreliosis. So if we go into our vaccine then, what is important to note is, because this is a vaccine that's not going to work as any other vaccine that you know, because OSPE is only expressed in the tick, in the midgut, when Borrelia is in the midgut on the tick, and during the feeding, when it takes up the blood, the expression of OSPE is downregulated. And when OSPE is downregulated, the Borrelia can move from the midgut to the salivary glands and into the vertebrate. This transmission takes at least 24 hours. So if you take off the tick within 24 hours, the probability of an infection is almost nil. Because most transmission starts somewhere between 24 and 48 hours. Which means that the vaccine is going to work in the tick. So when the tick is sucking up the blood, it takes also the antibodies that's going to neutralize and bind the OSPE and then block the transmission, and Borrelia is then eliminated in the tick gut. And this is then complement independent action, so you don't need complement for this. So they only need neutralizing or binding antibodies to have a functional vaccine. So based on what's known in literature, we have then designed our vaccine. So most common is of what I said, Borrelia fcalea in Europe. And there's been crystal structures from OSPE. There's serotype 1, but because of the sequence homology, the structure is going to be very similar for other serotypes. And the top, since this doesn't work now, yeah. In the top we have an N, there is an N-terminal of the crystal structure. This is a model. There we have the lipid moiety, because OSPE is a lipoprotein, and that's anchored into the membrane. And down we have a LA2, binding site for LA2 monoclonal antibody, which has been shown to correlate with protection. So if you're only cutting off then the right figure, the C-terminal part of the protein, then you have everything that's needed for protection, because the N-terminal part is embedded, because there's so many lipoproteins in the membrane that this part is kind of covered and not accessible for antibodies. So we are focusing then on the C-terminal, and what I probably missed there is what's interesting, because the C-terminal is protective. The problem is when you only use the C-terminal, it's not stable at physiological temperatures. So we need to stabilize it. So our bioinformaticians were then looking into where can we introduce disulfide bonds to stabilize the protein, and he came up with a long list, and then he made a priority where he chose 5 different sites where we then did all the modifications, checked for stability, immunogenicity, protections, and all things, and then end up with a B stabilization as our favorite. And to make a simple for a production of a vaccine, we wanted to have only 3 proteins where we fused 2 monomers with a 21 amino acid linker. And then we can end up with 3 proteins representing 6 different serotypes, which is more than 95% of the cases in Europe. And to get a high expression, we used the signal sequence from LPP, an E. coli protein, that we get the lipid moiety anchored to the protein, which is needed for the immunogenicity. And to test our vaccine, we have 2 different animal models. One is a tick challenge model, and the other is a needle challenge model. So we use 3 immunizations with 2 weeks interval. 2 weeks after the 3rd immunization, we either put on infected ticks or challenged with in vitro grown spirochetes. After 4 to 6 weeks, we take blood to do a serological assay to determine the infection, which is more or less the same as for human diagnostic kit, the VLSE ELISA. Or we take a part of an ear, extracting DNA, and do a qPCR. So at least one of these 2 assays has to be positive to get positive results. So the first question we were asking, does it matter in our heterodimers? Doesn't matter if a serotype is in the front of the heterodimer or in the back part. Because in the front you have the lipid moiety anchored, and in the back you would then anchor to the linker. And for the C-terminal part, in the front is linked, attached to a linker, and in the back part it's free. It's a normal. That doesn't have an influence. So we constructed two different ones with the serotype 1 and 2, and that we did because there we can test everything. We had the challenge model for both serotypes, and we can do all the tests. So we did then in our normal challenge model, we did then 3 immunizations and all things, and we checked then the ELISA coated with the serotype 1 protein, and then we see that if it's in the front or in the back, there's no difference. And if we're coating the ELISA plates with serotype 2 monomer, we can see that it doesn't matter if the serotype 2 is in the back or in the front. There's no statistically significant difference. Then we looked at the flow cytometry, the surface binding. Are there any difference in the functionality of the antibodies? Do they recognize the OspA on the surface different? And then we used either a strain Z7 expressing OspA serotype 1 or PKO with expressing OspA serotype 2. And we can see that both curves are overlapping. This will be done with pooled sera and different dilutions. So we don't see that there's any, that doesn't matter for their immunogenicity if they're in the front or back. And then we tested in our animal model against serotype 1 challenge in the needle challenge model, and we can see there's no difference. Or in the serotype 2, our tick challenge model, and we don't see any difference if it's in the front or the back, the serotype 2. So there we assume that it doesn't matter in which order we put the proteins in our heterodimers. And then if we compare our approach with others, because on the top is actually wild type OspA, which is the same protein as was actually used in the vaccine in US, the Lymerix. And we only marked here the dark blue is the part that we are using in ours. And then we have, with almost the same size of protein, we can encompass 2 different serotypes. And our competitor, Baxter, they have a different approach where they, in the C-terminal part, making a chimera of two different serotypes. So with the same similar dose, we have almost 2 times more of the C-terminal part when we're immunizing. So we think that we have an advantage there in immunogenicity and immune response. And there we are showing, we have immunized them with 3 different, the full-length protein, Baxter combo, which is then 3 different proteins, which has 6 different serotypes, and our combo with 3 different heterodimers with a 6 cell-based serotypes. And you can see that in most cases we are better than the other approaches, or we are equal. So superior or non-inferior to the Baxter combo in the immunogenicity. And the full length is always for serotype 6 coating, we have the serotype 6 full length and all the controls are similar, homologous. If we're then looking into protection, here is the tick challenge model. We have set up a tick challenge model for serotype 1 also by collecting ticks in the wild, put them individual ticks on mice, checking what serotype is in there, put on larvae on the mice, letting them molt to get the nymphs, and there the whole cycle is going. So it's almost a year's work to get one serotype into the ticks. And here you can show them with full-length OSPE, serotype 1, which is similar to the Lymerix, Baxter combo, our combo, and the placebo, that we have a nice protection when we immunize with 3 micrograms, so it's 1 microgram of each protein. You see that the numbers are a little bit different. We always start with 10 animals, but for serotype 1, because of the infection prevalence, we're putting on 3 to 4 ticks on each animal, and we only count animals where we have at least 2 ticks fed on the animals. Otherwise, if there's fewer, we don't expect them to be infected, so we don't count them. Even though some of these animals are infected, but we don't take them in because we have a set criteria. And we see that all 3 proteins are highly significant protective. And the same we have done also with serotype 2, where we also have a nice protection against all the proteins with a 3 microgram doses. We have done a lot of different animal studies, and we have actually the 2 tick models. We have 3 different needle models. So we can show protection against serotype 1, 2, 5, and 6. We now also in the lab have against additional serotype, which we are just waiting for verification. So in that case, we're only missing 1. And that's also the serotype that's the least common. So now we think that we have actually quite a good vaccine. So then we have done some upstream and downstream processes. So we have a semi-generic downstream process, and we do a fed batch fermentation in E. coli for the expression of the protein. And what we're also doing, as we heard earlier today, we do a phase separation with Triton X 114, because this is a lipidated protein, so it's going to go to the lipid phase. Our colleague, what we heard earlier, actually used it to trap the endotoxin, which means that we actually get a lot of endotoxin with our protein with this first step, but we get rid of a lot of whole cell proteins. So what we do in the hydroxyapatite column, 1 hour washing steps there, we wash it with Triton X 114 to wash away the endotoxin. So then we can actually get rid of it there. And we're ending up with a size exclusion chromatography, and with a normal biomass, we get what is 60 to 80 gram per liter. A purified protein, we get 2 to 4 milligram per gram biomass, and with a purity above 90%. What is triacylation? That's the lipid moiety, where we have three lipid fatty acids, and we have at least more than 85% of the protein does it. And endotoxin is rather low, and whole cell protein, whole cell DNA. And if we do a fermentation run with 11 liter, and assuming the same immunization dose as they had in Lymerix, we can actually end up with 65,000 human doses from a fermentation with 11 liter, which is quite good. That's going to make it rather cheap to produce. What is interesting is that the lipid moiety is important for the immune response of OspA proteins, because it was shown in Lymerix. They had some non-responders, and at Yale University, which is one of the study centers, they actually called back some of these, and they checked why are these people non-responders, and it turned out that all of these did not express TLR1 on the surface. And for that reason, there seems to be non-responders. So we're looking then, how does it look like, the lipid moiety in our protein? So we did a reverse phase separation in HPLC. So more than 85% of the proteins are triacylated, less than 10% are deacylated, which is just the one precursor form to the 3-acylation. And we also then choose one of the proteins, the serotype 5/6 containing heterodimer, to go on to do a GCMS, so looking at the fatty acids. And we have then C16, 61, 18 as the most common fatty acids, which has already also been shown when the full-length OspA expressed in E. coli with its normal single peptide. So it seems to be the same. So what we now have is then a protein-based vaccine that should theoretically protect against 6 major OspA serotypes, which is then prevalent in US and Europe. And we have shown in preclinical protection in our 5 different animal models, protection against 4 different serotypes. And we have the drug substance of an excellent quality produced in E. coli, scalable fermentation process. And in the fermentation, we get roughly 1.2 gram per liter lipidated mature protein. And with a similar generic scalable production, we actually have a high purity and a nice protein, which means that this product can then be ready for clinical trial within 12 to 15 months. Here you are showing acknowledgement the people is involved in it. So there are not only tick challenge, but with tick collections, we are doing other things also in the nature. So Andreas Meike is the head of research. Per Comstedt is not on the picture there, but he is heading the animal model studies. Wolfgang is our bioinformatician. Robert Schlegel is doing the downstream processing. Markus Hanner the upstream processing. And the others are technicians helping into the different animal studies, serology, flow cytometry, and all things. And also we have some collaboration with Sven Benström with the OspA protein. He was the first one to discover it then. And Hans Dausel in Berlin, we collaborate with him for the tick rearing. So he's doing that for us, and Volker Fingerle for strain collection. Thank you for listening. Yeah.
One is, I thought Baxter has gone out of vaccines now, so is there still a competition for you?
The last I heard is not yet sold.
That's what I heard as well.
Yeah. So we see. We don't know if it's going to be sold. They are ahead because they already done the phase 2. Yeah. But as long as it's stalling now, we can catching up.
Right.
Then, yeah.
Well, you could buy it yourself. That's another story.
We have looked into it.
The other question I had was, how are you going to do protection studies for clinical trials? Is there enough cases to do efficacy studies?
Yeah. Because if you're looking at the Limerick study, they had 10,000 subjects. But of course, you had to see what population you take into the clinical study.
Right.
That you're rearing it to. You don't want to have people living in the city
Yeah.
because they are rarely exposed. You want the people that's going out in the nature,
Yeah.
hunters, other hikers, and things like that. So you have to gear it to that region.
Not an easy clinical trial.
No, it's not. It's not going to be easy. It's not going to be cheap. The phase 3. No, that's true. Yeah.
Questions? I got one of them. I would like to know if you have several especially if you have used many animal models looking for correlation between borrelia burgdorferi and borrelia, not only serotypes related, in fact species related. So because there are clearly difference in the susceptibility between borrelia genus. So what is your
What in protection, or
in protection and in pathology that you can do?
no in pathology? But there are some studies, seen at least in human, that borrelia burgdorferi causing Lyme arthritis. That's the most common then in US. In Europe, we don't have it very much, Lyme arthritis, because that's not the most common.
For sure, but
Yeah.
you're stating that your vaccine is protecting you too in USA.
Yeah, but
So-
it's also protecting USA, of course. But since we also have serotype 1 in Europe, we have to protect against that one. And often the Garenia is the neurotropic strain. So they have it in humans then a little bit different tropism, but we don't see that in mice. We don't see that.
And did you
Yeah.
test it in macaques?
No.
Any more questions?
Yeah.
Thank you very much.